Okay, today we're going to talk about vaccines and the immune response. My name is Aimee Bernard. I'm an immunologist, and I teach biology, specifically immunology, at the University of Colorado, Denver. So vaccines are one of my specialties. So what we're going to talk about today is vaccines and the immune response. So to give you an outline of today's talk, I'm going to give you some background on the innate and the adaptive arms of the immune system and how they play separate yet interactive roles in the immune response. I'm also going to talk to you about specific cells of the adaptive immune system, such as B cells and T cells. And the result of activating T cells is immunologic memory. And memory is something we'll talk about that's really important to our response to vaccination. I'll also talk to you about passive versus active immunity today. And we'll talk about the immune system capacity, which, as you will see, is quite high. We can respond to a wide range and number of pathogens at any given time. And then I'm also going to discuss with you certain vaccines and how our immune system responds to different forms of vaccines. And I've chosen just 4 types of vaccines to discuss with you today. There are several more, but these are the types that you'd probably be most familiar with. And I'm also going to give them to you in the context of a common example. Then we'll also touch on the concept of herd immunity and how it's important for the majority of people in the population to be immunized in order to keep all of us protected. And then at the very last portion of the talk, I'm going to describe to you the efficacy or how well vaccines work, and then compare that to risks of vaccination. And this, of course, will be immune specific. Okay, so those are all the things we're going to cover in our talk today. These are just learning objectives. As a professor, I want you to all learn from this talk. So if you were to look back at this slide when the talk is done, these are things that you should be able to describe or explain or understand as a result of listening to this talk. Okay, so I was mentioning to you earlier there are 2 basic types of immunity, and we refer to them as innate and also adaptive immunity. And this is just a very simplified graph comparing innate to adaptive immunity. If we were to just, at a very basic level, define the differences between them, we would say that the innate immune system is nonspecific, meaning that the immune system does not respond to any specific pathogen. The innate immune system is really just our first line of defense, and it's not specific for any given pathogen. It responds to all pathogens in the same way. Now, in the context of adaptive immunity, that is a specific response. That is where certain cells of the adaptive immune system will respond to one specific pathogen, or actually a small component or piece of that pathogen. So if you were to compare and contrast innate and adaptive immunity, you would say innate immunity is nonspecific and adaptive immunity is highly specific. Also, if we were to compare them in the context of this table, you will see that there are differences in the context of the time it takes for the 2 arms of the immune system to respond. Like the innate immune system, again, which is nonspecific and our first line of defense, will respond within minutes or hours. If you compare that to the adaptive immune system, typically it's days. It will take days. And that's because the adaptive immune system has to recognize the pathogen that has invaded the body and kind of build an immune army to go off and defend itself against the pathogen. Whereas, again, the innate immune system, it's a built-in first line of defense. It's ready to go at all times, and again, nonspecific. And then we'll talk here about some of the major components. And again, I'm going to introduce you to the lymphocytes that play a role in the adaptive immune system, and those are the B and the T cells. So here we have an overview of the adaptive immune system. This is quite a busy, action-packed slide, but really what the basic message from this is that with the adaptive immune system, the major players are going to be the B cells and the T cells. So on the bottom of this figure, you can see the B cell. Now, B cells are cells of our adaptive immune system that have receptors on their surface, and these are the slightly Y-shaped structures you can see on the surface of this green cell. They will secrete antibody, and that's the receptor we see here on the surface, that will bind to and fight pathogen. So antibody is something that's produced by B cells to protect us against pathogen, and antibody will play a role in eliminating the pathogen from our body. Now, up above, we see two other forms of cells. These are both called T cells. T cells are the other major player of the adaptive immune system, and we have two forms, or two major forms. There's, on the left side, in blue, there are the T helper cells, and on the right side, we have cytotoxic T cells. They have two very different functions. T helper cells, just like their name suggests, they help other cells become activated and to participate in an immune response. So as an example, a T helper cell would help a B cell become activated and allow that B cell to produce antibody that would be secreted and then go off and fight infection. Okay? A cytotoxic T cell on the right side in orange, these cells, just like they sound, are toxic to other cells. So not healthy cells. They only go after like infected cells or pathogen. So because they are cytotoxic, they essentially destroy infected cells or pathogen. But combined, these cells are the major players of the immune response. So when we talk about vaccines and how our body responds to vaccines, what we're hoping happens is that B cells will recognize and interact with the pathogen, and you can see this down at the bottom next to the B cell, this little kind of oval with hair that says antigen. That could be something that would be found in the vaccine that a B cell would respond to and secrete antibody against if it remembers it. And T cells would also play a role in recognizing and remembering and then eliminating pathogen. So vaccines are going to be used as, or vaccines are given to individuals to be the first exposure to a pathogen. And vaccines will allow T cells and B cells to develop memory, which will allow them to remember that pathogen. And if they come into contact with the real pathogen, a second exposure, they will respond much more quickly and much more vigorously. Okay? So it's this, the combination of B cells and T cells, we need to produce memory so that we will respond much more quickly to a pathogen if we see it again, like in its real, live, and active form. So a couple of things that I want to talk to you about also are characteristics of the adaptive immune system. And again, when I refer to adaptive immune system, I'm talking primarily about B cells and T cells. And those are the cells within the immune system that are highly specific and that they are part of that adaptive immune system. But one of the characteristics of cells of the adaptive immune system, such as B cells and T cells, is that each cell in our body is essentially, its receptors are specific for different things. Okay, so as an example, if we see these 4 cells, and they're numbered 1, 2, 3, and 4, cell number 1 has a receptor on its surface that's specific for one type of pathogen, okay? Cell number 2 has a receptor on its surface that's specific for something else, okay? Same thing for cell number 3, cell number 4. We actually have millions of these cells in our body at any given time, and they are specific for pieces of pathogen, different things. So as an example, when antigen 2 comes into our body, because B cell number 1 is not specific, its receptor will not recognize and bind to antigen 2, it will not participate in an immune response. However, because B cell number 2, its receptor is specific for antigen 2, it's going to recognize and bind antigen 2, and this cell will be called into an immune response, or participate in the immune response. And also, cells 3 and 4 are not specific for antigen 2. They also will not play a role in the immune response. So this is to demonstrate that within our bodies we have millions of cells, each with a unique specificity, so that when a pathogen invades our body, it's likely we have at least one, if not a handful of cells that will recognize the pathogen and respond to it, but not all of them will respond. Okay, just those that are specific will respond. And this gets to the question that a lot of people ask me: when you immunize or vaccinate with several different immunizations at one time, aren't you overwhelming the immune system? And the answer is no. And that's because two things: we have tremendous diversity and we have specificity. So when I'm vaccinated with a particular type of vaccine, only a handful of the cells in my body at that time will even be specific for and respond to that pathogen. So this is, again, one of the characteristics of cells of the adaptive immune system. Another characteristic is memory. Again, this is the basis of vaccination. We want to create memory, T cell and B cell memory, so that when we encounter a pathogen a second time, we respond much more vigorously and much more robustly the second time. So the vaccine would be our first exposure, and we would build memory. And then if we were exposed to, say, a natural form of that pathogen, we would respond much more quickly and much more robustly. So this is a graph showing you, this is antibody level. Again, antibodies are secreted by B cells, one of the cells of the adaptive immune system. And on the x-axis we have time. So as you can see here, if you were to inject, and this is a mouse, with antigen A at time, roughly time 0, in order to start to produce antibody, your primary anti-A antibody response would take about 14 days. And again, the adaptive immune system, if you remember to that graph that I showed you earlier, or that table comparing them, it takes days for the adaptive immune system. It's because you're building an army of cells that are going to fight the pathogen. So it takes about 14 days for that primary response. Now, if we wait an unspecified amount of time, a couple of weeks, let's say, and we add back antigen A into the mouse, but at the same time we're also adding an antigen B, which the mouse has never seen before. This is the primary exposure to antigen B. We can compare a primary response, which we see in light blue at the bottom of the graph, to a secondary response, which is in a darker blue at the top of the graph. And if you were to compare the second response to A to the first response to B, you can see they're quite different. The secondary response to A is, you can see that it's quicker, it took less time to ramp up, and it's also much more robust. A lot more antibody is produced upon second exposure. And this is exactly what happens when you have a vaccine or a vaccination. Your primary response would be how you respond after the vaccine. The secondary response would be how you would respond in the event that you had a natural infection with the pathogen you were vaccinated against. So you can see the difference in time and also how much antibody is produced. And again, this is another characteristic of cells of the adaptive immune system. It's memory. So because I was just telling you about how memory is what we want to produce for T and B cells, I wanted to tell you a little bit about the differences in activation of a naive cell versus a memory cell. And here we're looking at B cells, but this would also apply to T cells. Okay? So as an example, for naive B cells, they require two signals. So you can see the green cell on the left, that's a B cell. I know that because that's a B cell receptor, that Y structure. They require two signals to become activated. Naive B cells require two signals to become activated. The first signal is always going to be through its receptor. So you can see this red little pathogen has bound to the B cell receptor. That B cell is specific for that red pathogen. That's going to be the first signal that's received. Okay? The second signal is where it gets help from the T cell. And that's where helper T cells come in. They help to activate other cells like B cells. So the second signal is going to be a form of protein-protein interaction between two molecules that are on the surface of B and T cells. And here we call them CD40 and CD40 ligand. So in order for a naive B cell to become activated, it needs signal 1 through its receptor, and it needs signal 2, which it receives from protein-protein interaction between the B cell and the T helper cell, that CD40 and CD40 ligand. Okay? Now, memory cells, because memory cells are actually naive B cells that have been previously activated and have participated in an immune response, memory cells can act a little bit quicker because when they bind pathogen a second time, remember they were naive at one time and they were activated, they only need signal 1. So a memory cell, as it's patrolling our body, looking for pathogen, once it interacts with pathogen through its receptor, it just needs that first signal, and it's going to be called back into an immune response. So it doesn't have to wait for T cell help because it already received help from the T cell back when it was activated the first time when it was a naive cell. So this is how memory is produced, and this is also why our memory response is so much quicker and also more robust. Okay, so now that you have a total understanding of memory, I'm going to talk to you a little bit about the two major forms of immunization. The two are passive and active immunity. And passive is just what it sounds like. Passive is when you are not asking the immune system to participate in immunity at all. You are receiving passively, typically antibodies from another individual, antibodies that were produced by someone else and then pooled and given to you, or it could be from the mother would pass them through the placenta to the unborn fetus. So this is passive transfer from one person to another. Again, typically it's antibodies. Passive meaning the immune system is not called into play. Active, on the other hand, is when pathogen exposure generates an effective immune response. You are asking the immune system to participate in a response. So here, this will lead to memory of the pathogen. So as an example, as you can see here, vaccines are an active form of immunization. Your immune system is participating in a response. And you can also see that natural infection would do that as well. So a natural infection with a bacteria or virus. But vaccines, again, a large portion of the active immunity. The goal of active immunization is to produce memory and long-term protection. One thing you need to know about passive immunity, or the transfer of antibody, is that it's transient. Okay, it's just short-lived. For however long those antibodies will survive in your body is as long as that immunity, passive immunity, will last. However, with active immunization, if you produce immunologic memory, this will result in long-term protection. Memory cells are thought to live at least 30 years or longer. So we have memory cells that will patrol our body for years and years, hoping to come into contact with that pathogen again. And again, this is because T cells and B cells of the adaptive immune system have been activated and are patrolling our body, waiting for a second or subsequent exposure to the pathogen that they were built on. Okay? I want to do a quick concept check within the audience here. This is my chickenpox concept check. What I usually do is ask people in the audience if they have had chickenpox to raise their hand. Okay, so most people raise their hands that they've had chickenpox or that they've had the chickenpox vaccine. And then I ask people to raise their hand again if they had chickenpox a second time. And most of the time people do not raise their hand. So the question that I have for you is, do you think that you only came into contact with the chickenpox virus one time in your whole life? Or do you think that you've come into contact with chickenpox several times throughout your life, and that because you had memory, you may have actually become infected, but you responded and your immune system took care of it before you were sick? So the answer is B, right? We've come into contact with chickenpox many times throughout our life. The fact is we have memory cells that remember and respond before we become ill. Okay. I also wanted to talk to you about the capacity of the immune system. And this is an analogy that comes from Dr. Paul Offit. And he comes from the Children's Hospital of Philadelphia. And he is very well known in the vaccine literature. He's a vaccine advocate. And this slide, actually, I have to give credit to Dr. Rachel Herlihy. She's at the CDPHE here in Colorado, and she let me use this slide from a talk she had given previously. But really what this tells you is that the amount of antigens that we would be given from vaccines is really a drop in the ocean of all of the pathogens we would encounter in our lifetime, and also the amount of pathogen that we could potentially combat at any given time. Because we have this tremendous diversity and we have this tremendous capacity of the B cells and T cells in our body to respond to hundreds to thousands to millions of pathogen at any given time. So it's really just, he likes to call it a drop in the ocean. And then this also comes from Dr. Rachel Herlihy, a talk that she gave, just showing you shots versus antigens. And this is over time, so from the 1900 to 2000. And what you can see, if you look at all the vaccines that are recommended, the total number of proteins or polysaccharides or just bits of pathogen that you will be exposed to is right around 125. So again, that's just a drop in the ocean compared to how much we could potentially handle and how much our immune system could combat at any given time. Again, we have millions of B cells and T cells in our body every day, all day long. And these are all the number of antigens that you would have as a result of following this vaccination schedule that's recommended. Following that slide, I wanted to talk to you a little bit about the diversity of the adaptive immune system. I know I've mentioned this a little bit as we've been going through, but this is another characteristic attribute of the adaptive immune system and the cells within. There is tremendous diversity in our body at any given time. Every B cell in our body, every T cell in our body, they have unique specificities. So we have 10 to the 9 to 10 to the 11 different antibody specificities in our body at any given time. So we can handle thousands of antigens at one time. Now, of course, we're probably not going to do that, but we have the capacity to do that. So due to the tremendous capacity of the immune system and also that specificity that's built in with each cell being specific for something different, there's no physiologic reason to design an alternative schedule or to fear that you will be overwhelming the immune system by getting 4 or 5 shots or vaccinations at one time. You will not overwhelm the immune system. The capacity of the immune system is absolutely tremendous. It can handle a lot more than we actually give it. And the other thing I wanted to mention too was, and this also goes back to Dr. Offit's ocean analogy, we come into contact with so many pathogens every day that we just would normally inhale or ingest or touch, that we're fighting off pathogens on a daily basis that we don't even count. So vaccines are obviously something we can count how many antigens we're responding to, but we respond to hundreds of them a day, just in daily contact that we don't count, we don't see them, right? So we're responding to more things than we would even know just on the basis of living and interacting with things in our environment. So now I'd like to talk to you about, now that we have kind of a solid basis of all of the adaptive immune system, we're now going to talk a little bit about vaccines and the immune response. And I've chosen, again, 4 types of vaccines I'd like to discuss with you, and then an example of each of these vaccines that you're most likely familiar with. So we'll talk about live attenuated, also inactivated vaccines, conjugate vaccines, and subunit vaccines. And we're going to talk about them in the context of how the immune system responds to these types of vaccines. Okay, so our first we're going to talk about is live attenuated. And the example I give for you is the nasal form of the flu vaccine. So if you take that nasal form of the flu vaccine, this is a live attenuated. And what that means is that it's a live virus, but it's weakened. So attenuated just means weakened. And it is unlikely to cause disease. It's weakened to the point that it's no longer able to cause disease. And these weakened strains are derived from or come from what we call the wild type or virulent strain. But again, they are weakened to the point that they do not cause disease. However, in order for them to be effective, there are a couple of properties that these types of vaccines have to possess. One of them is that the antigens on the surface of the virus have to be the same or very similar. Because when the immune system, when cells of the immune system come upon a virus, they are going to recognize that outer component of the virus, the things on the outside of the virus. You can think of it like as the coat of the virus, the coat that the virus is wearing. That's what the immune system is going to recognize and respond to. So if the coat looks different, it won't respond. So you want the outer portion of the virus to look the same. We say that it has to molecularly resemble that wild type virus. So it has to look similar enough for the immune system to respond to it. And then obviously that wild type virus has to be attenuated or weakened to the point that it has little or no virulence, that it will not cause disease. So those are 2 of the properties that they have to possess. Now, as far as the immune response to attenuated vaccines, this is probably one of the most effective types of vaccines that you can have. And that's because you will actually be infected by virus. It's live, and it will replicate. In this case, it will replicate in the mucosa of the nose. And as a result of that, the virus will increase in number. It's going to replicate. And this means your immune system is going to have more viral material to respond to. So that's going to provoke or induce a pretty wide-ranging immune response. So as a result, the immune responses typically will include B cells as well as CD4 helper T cells and CD8 cytotoxic T cells. So all arms or all components or major players of the adaptive immune system are typically involved in our response to live attenuated vaccines. So this, again, live attenuated vaccines are usually very effective. Another type of vaccine that you're most likely familiar with are the inactivated virus vaccines. And the example here is the injection form of the flu vaccine. So if you get the shot form, you're getting an inactivated form. And what that means is that the virus has been inactivated or killed. There are a couple of different ways in which they will inactivate or kill the virus. Sometimes it's a chemical treatment like formaldehyde, but either way, the virus is no longer living. So it's not going to infect you and replicate and make more. Whatever is in the shot is all that you get. Okay? Of course, there are challenges in determining correct concentrations of chemicals and making sure that you inactivate it completely, but this is something that is dealt with by research and design and through safety trials has been deemed effective. Okay, so vaccine biologists, you need to inactivate the virus, but you don't want to destroy it beyond recognition. So if you treat it with a chemical to kill it, you want to make sure that it still resembles the wild-type virus so that if the immune system comes upon this after seeing the vaccine and building memory, those memory cells, when they come upon the wild-type version, if you were exposed to flu, that they would recognize and remember and call into play that memory response. Okay, so you want to make sure you don't destroy it beyond recognition. So our immune response to inactivated vaccines is quite good as well. But because the amount of antigen that's in the shot is all that you will get, your immune system will have less to work with. It's going to work with whatever's in that single dose. Okay? As a result of that, inactivated vaccines typically only induce B cells, meaning that we see antibody. In humoral, this is a term that immunologists use when they're referring to antibody and molecules that are released into the humors or the fluid, in this case the blood. Humors is a very antiquated term that we use for like fluid of the body. So humoral antibody response, that's that B cells have been activated, but typically these types of vaccines don't call into play T cells to the same extent. We usually see B cell memory, but not T cell memory. That is variable, but this is a typical response. This is a question that I often get from a lot of my students is, well, why do I need a flu vaccine every year? So one thing I wanted to just briefly talk to you about was influenza viruses are constantly changing. One of the things that viruses do best is to continually mutate. So we see new strains emerge each year. And there are collaborating centers around the globe that will type different strains every year. And then the three most common strains, 2 strains A and 1 strain B, are going to be included in the current vaccine. So the way that the flu virus changes is through something called antigenic shift, which is a major change in some of those antigens that are on the outside of the virus, or through antigenic drift, which is a minor change in surface antigens. Either way, both of these can alter what's seen on the outer portion of the virus, and that's why we need to have a flu shot every year, is because the flu virus is very good at changing what it looks like. So we have to make sure that we are vaccinated to respond to the newest version or strains of the flu that are coming around and emerging every year. Okay, conjugate vaccines. This is another common form of vaccines. The example I'm going to give you here is Hib. This is Haemophilus influenza type B. It's a bacterium. So Haemophilus influenza is a bacterium. This is a conjugate vaccine. So if you look at this upper left of the drawing or this slide, you can see that this Haemophilus influenza is coated in polysaccharides. And polysaccharides are kind of like carbohydrates that are on the outer portion of this bacterium. Well, one thing that's kind of important to understand how the immune system responds to this is that T cells only see protein. T cells don't see carbohydrates or polysaccharides. They only see protein. So when you see if there's a pathogen that's coated in polysaccharide, T cells will not respond. And we need T cells to have a wide-ranging immune response. So the way that vaccine biologists have worked their way around this is they've come up with something called a conjugate vaccine. And that's where they will take the outer polysaccharide coating of the bacteria and they will link it to a protein. So they will conjugate the polysaccharide to a protein, and this protein component is what's going to allow T cells to respond. So the protein is going to call the T cells into play. Now B cell receptors can respond to anything. They can respond to polysaccharide, to protein, to lipids, as long as their receptor is specific. Remember, each B cell has a different specificity, each B cell receptor. But some B cells in our body and their receptors will be specific for polysaccharides. So this conjugate vaccine is going to call into play B cells that would potentially respond to the polysaccharide, but also call into play the T cells that would respond to the protein. So this conjugate vaccine was developed so that T cells would also be called into play and make a much more robust immune response. So this is kind of just a text version of what I just explained to you. B cells can recognize all types of antigen, proteins, polysaccharides, lipids. Again, their receptor has to be specific for it. But B cells combined can recognize just about any molecular component. T cells, we know, will only recognize protein. So in order for B cells to get T cell help, you're going to have to activate T cells and call them into play as well. So this protein conjugated to that polysaccharide is going to allow for that to occur. Okay? And also B cells and T cells have to recognize epitopes. That's a fancy term for like a piece of antigen or an antigenic determinant from the same molecular complex, and we call this linked recognition, which I'll show you in just a second. But really what this boils down to is that B cells and T cells work together. Okay, so B cells and T cells work together. And again, this conjugate vaccine is allowing B cells and T cells to work together to respond to things like Haemophilus influenza type B. So this is linked recognition. And this is showing you why we do this in the context of vaccines. So I'm going to walk you through this slide. If you look at the top left panel, you can see that in yellow is this B cell. And on the surface, that's the B cell receptor. We'd also call that a membrane-bound antibody. Okay. Here we have, this is a conjugate. We have in blue, these are little pieces of carbohydrate, and it's linked to or conjugated to this red ball that's a protein. Okay? The B cell receptor, that receptor is specific for this blue carbohydrate. It's going to take that whole pathogen, or in this case conjugate, inside the cell. And what it does then is it breaks it down into pieces. And those pieces, as you can see on the second panel, bottom left panel, these pieces are going to be broken up, and this molecule here, that's called MHC protein. It's really a display protein that certain cells of the immune system use, including B cells. This MHC, or this display protein, is going to pick up different pieces of the broken down pathogen, which is this carbohydrate and protein, blue and red, and it's going to now display all the bits on the surface, all the pieces that comes from this carbohydrate and protein. And as you can see on the top right panel, the MHC is displaying this little piece of red, that's the protein, and the T cell, this is a helper T cell in the top right. That T cell receptor is specific for that little red ball. It's going to recognize that little piece of protein. because of that, it's going to give the B cell help. It's going to help to activate it. Remember, naive B cells need two signals. Signal one is binding pathogen or antigen through the receptor, and the second is that CD40 ligand interaction between B cell and T cell. So it's getting T cell help. That's now going to activate this B cell. This B cell can now go on to produce lots of antibody to fight off the pathogen. And if you notice here, it's making antibody against the original blue polysaccharide or carbohydrate that it responded to begin with. So this B cell will make a lot of antibody to respond to that pathogen. But where linked recognition comes in is that the B cell receptor and the T cell receptor that gives it help have to recognize something that were at once linked, that linked recognition. So even though the T cell is recognizing a different part of this complex, it was still linked. Okay? So the B cell responded to the blue polysaccharide, whereas the T cell responded and gave help based on recognizing that little bit of protein. So this is where vaccine biologists can kind of manipulate or regulate our immune response by conjugating or linking these two things together, allowing for T cells to give B cells help. Okay? And we see that, so we would see this in response to a conjugate vaccine. So this is how the immune system would respond to those conjugate vaccines. And as you'll notice, the B cell makes antibody to that carbohydrate piece that it recognized to begin with, not to the red bit the T cell receptor bound to. That's really just to get help. That's to increase its chances of help. But it's going to secrete antibody to the carbohydrate. It's going to clear the pathogen, and then that pathogen will be removed from the body, and the immune system will have defended itself against that pathogen. So this is really how the immune system responds to conjugate vaccines. This is just a graph showing you the rate or the incidence rate of invasive Haemophilus influenza type B. On the y-axis, on the x-axis, we're looking at time. And you can see that once the vaccine was introduced, that the rate or the incidence rate of infection dramatically decreased. Haemophilus influenza B can be a very serious illness. So having this drastic decrease was quite good for the medical community. Okay. So another type of vaccine we'll talk about, and this is the last type of vaccine, are subunit vaccines. And the example that I'm going to talk to you about is the acellular pertussis vaccine. Now, what you see in this slide, this comes from a virology textbook, and this is looking at the basic formulation for the influenza vaccine. But the same principle of how this is made could apply to the acellular pertussis vaccine. And what happens in a subunit vaccine is that instead of giving a whole pathogen, you can give fragments of the pathogen to trigger an immune response, so just pieces of the pathogen. So the subunit vaccine will contain purified components of the pathogen that are then combined to make the vaccine. So again, this is not a live pathogen. This is just pieces of the pathogen. And typically, we pick pieces of the pathogen that are on the outside, again, kind of like the coat that the pathogen would wear, the things that the immune system would recognize and respond to. So only the outer surface antigens are used for subunit vaccines. And if you look at, depending upon the company and the formulation for the acellular pertussis vaccine, these subunit vaccines can contain some of the following antigens, but it depends upon which manufacturer. But these are all components of Bordetella that are found on the outside of that pathogen. So they use things that the immune system would recognize on the outer portion and then respond to. So our immune response to subunit vaccines, because they're pieces of pathogen, they're typically not as immunogenic, meaning they do not induce as robust of an immune response as whole pathogen. So typically the response to these types of vaccines is much lower than it would be for, like, say, a live attenuated. And that's why if you get a subunit vaccine, you typically need booster doses. So booster doses are often needed just to kind of prime the immune system even more and hopefully induce more of a response. So subunit vaccines can be quite effective, but typically they're only effective when you have several doses. So as an example, if you were told that you needed your original shot and then 3 booster doses, you need to do all of those to be protected. If you just get the original shot and 1 booster dose, that's not going to protect you as much as getting the entire amount of booster doses that you need, because through studying efficacy, it's been shown that a certain number of booster doses will allow the best possible immune response. And one last thing I wanted to say about subunit vaccines is that the choice of antigen is often limited because you have to use what's on the outside of the virus or the bacterium. So you're limited to what that pathogen provides you on its outer coat. So that is somewhat limiting to what will be used by vaccine biologists. Okay, now that we're done with vaccines. Herd immunity. This is kind of an important concept in the vaccine and immunology world. So if the majority of the population, or the herd, is immune to an infectious agent, the chance of a susceptible or unvaccinated individual contacting an infected individual is so low that that susceptible person is not likely to become infected. That's kind of the concept of herd immunity. And those that are vaccinated will protect those that are not vaccinated, because we will not see illness or sickness or spreading of disease. And I put an asterisk next to this not vaccinated because there are people actually in our community that cannot be vaccinated. So it's not something that we're talking about a choice not to be vaccinated. There are some people that actually can't. So as an example, there are some infants that are just too young for a particular vaccine. Their immune system is not matured enough to respond appropriately, so they do not get certain vaccines at a certain time. There are also people in our community that are immunocompromised and not able to be vaccinated. Examples are people that are HIV positive, so they have the HIV virus. Those that are undergoing chemotherapy, they are also not, they should not have vaccination at that time because they are immunocompromised, meaning that their immune system is weakened and not working up to par. Also, there are individuals in our community that are immunodeficient, meaning that certain parts of their immune system do not work well enough to respond appropriately to a vaccine, so they cannot be immunized. And then also transplant patients. Transplant patients are given immunosuppressants so that they will not reject the transplant, but as a result they're also immunocompromised, so they cannot respond to infection or pathogen safely. So these are the people that we need to protect. Those of us that can get vaccinated should get vaccinated so that we can protect the people that actually can't be vaccinated for medical reasons. This is just a graph looking at measles and measles vaccine. And what you can see, if you look at the number of cases compared to the year, the vaccine was licensed back in, is that 62, 63? And you can see a dramatic decrease in the number of measles cases as a result of vaccination, suggesting that vaccines work quite well, and that when the community is vaccinated, you do not see cases of measles. And what the inset shows you here at the top right is that it's kind of blowing up this far right portion in the 80s, is that when there are people in the community that decide they don't want to get vaccinated, we will see outbreaks or epidemics of certain infectious diseases that will reemerge as a result of a breakdown in herd immunity. So this can become quite serious, because if you have a breakdown in herd immunity, now measles, in this case, will be spread, and those that can't be vaccinated, like immunocompromised or HIV positive or those undergoing chemotherapy, are susceptible and could potentially pick up the disease. And measles, you can die from measles. It's a serious disease. Okay. So I want to talk to you a little bit about potential risks of vaccination. Obviously, a vaccine is a medicine, and like any other medicine, it could cause a reaction. However, the chance of a vaccine causing serious harm is really very small. And any vaccine can cause side effects. Most of them are minor. So you might have soreness at the injection site. You may have a low-grade fever. This is normal. The way that our immune system works, you want cells to come to that area. You want to have soreness at the site. You want to have a little bit of swelling, maybe a little bit of a low-grade fever. That's telling us our immune system is working, that there's an immune response being produced against the thing we are becoming vaccinated against. So that's quite normal. Not having that would not be normal. So if you wanted a complete list of potential risks, I would suggest you go to the CDC website. The CDC website is an amazing source of information for disease in general, but also vaccination. So again, in the context of immune response, as far as what happens as a result of vaccination, swelling, redness, and soreness, again, that's good. Our immune system is working. Cells of the immune system are being called to that injection site, and they are going to respond to the pathogen in the vaccine. And again, another thing that may occur is fever. And again, this is likely due to activation of immune cells and the release of particular chemical mediators that help that immune response move forward. So these things are all normal in the context of an immune response. Another disorder that I wanted to talk about briefly also is called Guillain-Barré syndrome, mainly because when I teach about vaccines in my classroom, a lot of students will ask about Guillain-Barré syndrome, or GBS. And really, the bottom line of what I have to tell you is that it's more likely to develop GBS after a natural infection than after receiving a vaccine. However, they're in very rare cases, and this is back in the 1970s, there was shown that after a particular flu vaccine that there was a small increased chance of GBS. Now, this was only one more case per 100,000, so it was quite small, but this is often something that people will come back to. But again, I want to stress it's more likely to develop GBS after a natural infection, and you can see most of the times it's after an infection with this particular bacteria. So there are particular illnesses that are linked to the development of GBS. And also, just to give you a little bit of background, it is a rare autoimmune disorder. An autoimmune disorder is when cells of your own immune system will attack self. And what this will often result in is damage to nerve cells that can cause muscle weakness and sometimes paralysis. Another thing to think about is most people recover fully from GBS. Now, symptoms can last for a few weeks or maybe months, but usually they recover quite fully. Either way, it's something I wanted to just share with you because this is often a question when talking about vaccines. Okay, the other thing I wanted to talk about was this is a blog I wrote for the Colorado Health Foundation. Obviously, I'm a vaccine advocate. I'm an immunologist, married to a pediatrician. We vaccinate. Yes? I have twins. We vaccinate them. One thing that I would like people to think about is that when vaccines work, we don't see anything. So when people that have, we see measles and polio and these horrific diseases, we saw those primarily before the vaccine was available. Now that vaccines are available, we don't see that. So the general public hasn't seen horrific diseases in quite some time because vaccines work. So when vaccines work, the outcome is not as dramatic as when you don't get vaccinated, because we don't see these anymore. We're very lucky. In the United States, we're very lucky that we have all of these vaccines so that we rarely see diseases, these infectious diseases that can result in disfigurement or even worse, death. And these are the references for my talk. So if you wanted to learn more, I would look at a couple of these textbooks. I would refer you to the CDC website. That's where I got a lot of my information. Other than that, thank you. Yay. That was 1 hour exactly.